Good morning, everyone. I'd like to begin by thanking you all for taking the time to join with us today for what should be an interesting talk. But before I introduce our speaker, I would like to briefly touch upon some housekeeping guidance for the webinar. Firstly, the webinar will be recorded. During the presentation, there will be an opportunity for questions to be asked. If you could do this via the Q&A section of the application, that would be wonderful. Your questions will only be visible to yourselves and the Greiner moderators. We will try to answer as many of the questions as time allows at the end of the session. Any that we don't manage to get to will be managed and answered offline. I would now like to introduce you to our speaker, Dr. Sven Muehlfriedel. Sven is Greiner's Global Product Manager for our sample storage portfolio and brings both a wealth of technical manufacturing expertise and a practical biobanking knowledge to us all. I will now pass you over to Sven. Thank you very much.
Ladies and gentlemen, I would also like to welcome you from my side and thank you, James, for your kind introduction. So the title of today's webinar is Good Practice in Biobanking, a Labware Manufacturer's Perspective. I want to have a quick view on the agenda first. I want to start with some words on workflows and how appropriate labware can be chosen with regard to workflows. I want us to talk about and think about sample tracking and how this can be made secure and what our options are for sample tracking. I want to continue with process automation and finally spend a few words and have a few slides we can look at about quality standards where we can go a little bit more into detail. Sample storage workflows, and here exemplified based on the biobanking workflow, often share common themes. Like at the beginning, a sample is drawn or collected. Afterwards, there's a processing or aliquotation step. In the center portion, we see that, of course, the actual storage stage is happening. Afterwards, we retrieve and distribute samples, and at the end, samples are going to be analyzed. In sample tracking, but we have also a whole other world of information which we also need to deal with. This is shown down here. This can range from information about samples themselves, about processing information, to storage conditions, and finally end in analysis results. And the point we want to make with this first slide, which shows the workflow, is actually that at the link between the material flow, the sample, and the information, we find our storage vessel, here symbolized and exemplified based on a storage tube. It's a Greiner Bio 1 biobanking tube. And it's important to consider that also this item and not other items only are very quality critical for the entire process, because the storage container actually contains the sample for a very long time, so it's in direct contact, but it also carries the identifier, which gives the sample an identity, which allows for all this tracking and tracing. And if this would be gone, the quality of your sample, or actually your entire sample, would be lost. So taking this into account, it is very important that a storage task is very carefully described and defined. Any sample storage task can only be as good as the definition of its purpose and downstream applications are. And this brings us to intended use statements concerning your biobanking or sample storage approach, and also the intended project you actually want to carry out. This sometimes starts a little bit at the end of the workflow, where we wonder ourselves, what actually will we do with the samples at the end? Who will be the scientists who take advantage of the samples and do their research with these samples? How will they be shipped, and what will happen to them? Starting from there, we go back and say, okay, if this is the intended purpose of our project, then this would be the amount of samples we need, the quantity and quality of samples we need. And the samples must be fit for purpose, so that the whole project makes sense. It's a different story if you offer, let's say, cells for DNA isolation, and this is the purpose of your project, or you offer cells or serum to analyze biomarkers in it. That's a whole different story. And if all this has been clarified, then we finally come to the last group of items that need to be fixed, and these are actually your storage containers, your storage equipment, equipment you use for processing, but also other characteristics of your project, such as which temperature do you want to store it, and also for which duration in time do you want to carry out storage. I want to give a quick overview on what items, what vessels, containers are out there today for storage purposes. And they are on this chart put in context to different types of storage attempts. Of course, the biobanking is an important one here. It starts in the left upper corner with these rather classic cryotubes. Often they are in a 12 millimeter diameter. 81 or 100 of these items fit in one box. You know, these very classic cardboard boxes. That's a very common item that is used, but also these 81 and 100 weight boxes that come in plastic. And these classic cryogenic tubes are very often used for cell or microbial banking, but also for biobanking purposes. But as soon, and we will hear more about this later on, as soon as we see an increase in throughput of samples or demand of samples, and maybe also an increase in automation that is used in a biobank, these items are often used with machine-readable identifiers. We talk, excuse me, we talk about barcodes here or data matrix codes. The items themselves, the storage tubes, are pretty much the same, but they have in addition these barcodes and data matrix codes. Often still they are used in these 81 and 100 weight boxes. But if indeed automation is gaining importance and throughput is really getting high, then there's definitely a trend towards 48-way format racks, which you see here in this picture, because these are automation compatible. And also about this, we will learn a bit more on the upcoming slides. And then another topic and another common theme is getting smaller, storing smaller amounts of samples. So other containers gain interest now. The principle is pretty much the same. You have these racks, you want them to be automation friendly now, but the tubes are smaller. And since mainly the diameter is changing, also a little bit the height, you can now accommodate 96 of these tubes on the standardized footprint. We now talk about liquid biopsy biobanking, which is very often carried out in these formats, but also compound storage is the topic here, because compound storage sometimes is done in such small tubes in these automation-friendly racks. And then to make the story complete, there's also storage plates out there. They start with 96 wells per plate footprint, and then they go also smaller. A 384 well format is absolutely common as well. In this field, storage plates are very compact and very good compound storage vessels. The only difference between the aforementioned 96 weight tubes is that you cannot cherry pick samples. So different formats, different aspects of storage, and a trend towards automation and getting into smaller volumes. And this storing in smaller volumes is actually one aspect I want to now spend a slide with, because I find it's important to think about it and also deduce some good practices from it. It is the fact that we should think about the volume of a sample we need at the very end. An end user, a researcher, or whoever is carrying out an experiment requires to get his data. And this amount of sample should be, in the best scenario, used in one shot. Because if you, let's exemplify it, store 2 milliliters of serum, yeah, but at the end, you know, your end users taking advantage of your samples will only use 50 microliter of serum, then it's not a good practice to store 2 milliliters and then always thaw the sample, take 50 microliters away and refreeze the rest. Because what will simply happen, and we have a lot of literature today and a lot of findings that substantiate this hypothesis, what will happen is your biomarkers will degrade. And today we know that just one thaw freeze cycle too much may already destroy your samples. So the conclusion is, again, you have a biobanking approach, you think about your intended project and also your fit for purpose samples. Store your sample type in the best and most appropriate format and volume. And examples are given here. It's pretty much a standard to store cell stock and biopsies in 1, 2, or 4 milliliter samples, whereas the 4 milliliter volume preferably is used for saliva and urine. These materials don't have too much material and cells and DNA in it, so that you require larger volumes. But everything else, serum and especially also DNA samples, they really are absolutely sufficient if they come in very, very small single-use aliquots. It's very common to store 250 microliter of DNA today and then hand it out to the user for an endpoint analysis. And for serum, it ranges somewhat from 500 to 1,000 microliters. It's also important, there's a little note down here, that you respect working volumes, especially if you store in the cold, then you should respect working volumes, not go above, because if you fill the tubes too much, tubes can get destroyed and you would lose your sample. I want to continue with another concept and step that happens very early in your workflow, namely you have to close your tube at a certain time. Here, mainly 3 different solutions are existing today and represent our state of the art. On the left-hand side, we see the heat sealing approach. So here it's either a plate or a rack with tubes in it, and you have a foil which you put on top, then heat is applied for a very brief moment of time. Then a layer's melting is forming a seal, and there's even options today when you remove the carrier material, then you end up with individually sealed wells or tubes. That's possible. So this option allows for single use. It allows for temperatures down to -80 degrees Celsius, but the system is not reclosable. A very convenient system though. In the center portion, we see silicone push caps. Silicone push caps are also a very commonly used solution for closing tubes. You can also find solutions for plates. They are like little, as it says, silicone or rubber push caps you push into the top of the tube and they form a seal. They are reclosable and they can also be used down to -80 degrees Celsius. We did not make good experience with these solutions if users wanted to go further down and store in the vapor phase of liquid nitrogen. Finally, last not least, on the right-hand side, you see screw caps. I think we can suggest that there's a little bit of a switch in paradigm or a change, namely in a way that more and more biobanks and also other stores, compound stores, use screw caps now. I think the reason is given here by the many features and advantages such a system is providing. First of all, it's reclosable. It's suitable for any storage temperature, so it can be used for vapor phase storage above liquid nitrogen. And also, and this is, I think, a striking and important point, it can be used for air shipment of samples. You see this if you check the box or the paperwork certificates from your tube supplier providing a IATA statement, IATA certification. That's a special test that is performed, and it assures that samples can be shipped with the airplane. And now I want to go a little bit back to what I said at the beginning. Think about your workflow and think about every stage and make a good choice of labware. This is, for instance, one point. If you want to send samples out by air, and often I think this becomes a necessity, then screw top tubes or any kind of solution that is provided with the IATA certificate is an optimum solution. If you don't have this, then you have to circumvent the thing, and this usually causes problems. I want to spend one more slides with these closures, but now focusing on the screw top solution in particular. On the left-hand side, you see a video up here where a 96-way layout of tubes in racks is shown, and you can see how a little picker is used to pick up screw caps from the cap carrier. From Greiner Bio 1, it's actually a regular 96-way plate that is used as a cap carrier, and it's a very convenient solution because you can individually close the tubes, you get a good grip and hold on the tubes, and you even get a good idea how much you have tightened the tube to produce a good seal. A very common approach, often used, but you may run into a stage of your biobank or project, if it's not from the beginning on, where throughput is really getting higher. Actually, I guess this is also what one wants. One wants really to get samples in and work with them. And then you may need to think about other solutions like semi-automated or automated solutions. In the center, you see such a semi-automated solution. Again, it's a device from Greiner Bio 1. It's our Cryos decapper. It looks a little bit like a multi-channel pipette. Sometimes people from the distance think, oh, wow, you have a multi-channel pipette there, but it's actually a decapper. The thing is that it has 8 different spindles. They have collet spits down there, and each of them is driven by an individual electric motor. So if you push the decapper and its spindles into the screw caps of the tubes, it interlocks with the screw caps. You push a button, you cannot see it in the video, it's in the would be in the upper part of the video, and then it unscrews the tubes and opens them. You can park the screw caps again in a 96 well plate or any cap carrier your supplier would supply, and then do your pipetting job and reapply the screw caps by just pushing the other button. The good thing is also not only that it's semi-automated and assists you and helps you to speed up in your process, but it also closes the tube with a very, very defined torque value. In our example, it is 6 Newton centimeter. Every supplier should support you with a nominal torque value because you want to make sure that you are not closing your tubes too loosely or you over tighten your tubes. In our case, it's 6 Newton centimeter. And the device is really very precise. And this brings me to the idea that it's rather, I can really suggest already a common and best practice that if you want to store longer, I suggest a year, 2 years, or really look at long-term storage, which can be 10 or 20 years, then there is such a phenomenon like water evaporating out of your tube. When we were first confronted with this, we were surprised because we thought at these low temperatures, this doesn't happen, but it's happening. But the more precisely you close your tubes, you are safe for long-term storage because water evaporating from your sample will be really, really covered. It will be a very, very low percentage of water that can enter the tube. So it's the best that you can do to preserve your samples for a long term if you use such automated and assisted devices for tube closure. On the right-hand side, we see finally the fully automated solution exemplified as a device from Hamilton Storage. The principle is pretty much the same. There are these little spindles and collets and there are motors. The system here opens row or column wise, uses 96 well plates to store screw caps in between liquid handling steps, but there's also other devices which open all tubes at once. They hold the screw caps, you get the plate out, you can do whatever you need to do, and you close again. And again, here we also have this very precise closure, which I already made the point is so important for long-term storage. Now I want to step away from this like aliquoting and closing the tubes, these very, very early steps in your workflow, but I want to move over to sample tracking, as I told you at the beginning. It is strongly suggested, and it's getting more and more a common practice, and you even find it in the currently published norms and ISO standards for biobanking, that if possible, you should use machine-readable identifiers on your tubes. I already mentioned it earlier. They can be linear barcodes or two-dimensional barcodes. They provide you with a whole lot of advantages. 3 of them are listed here. First of all, you are very error-proof and you are automation friendly because it is machine readable. The integrated scanner can read it and no human being is involved any longer to like read an ID and type it into a computer system. Or let's even think about labels with handwriting, which is even worse. It also allows, the system also allows for sample anonymization, which is often a must in a biobank. If you have consent and so on, you need to anonymize your samples. And as soon as you have scanned the barcode and have entered it into the system with all the information that is still allowed, you are done with this step. And also, what is important, these barcodes are supplied from manufacturers in an unambiguous way. So each of these tubes or plates carries an identifier, which, if we talk, for instance, about Greiner Bio 1, is absolutely guaranteed to be unique. There is no other item you can buy from Greiner Bio 1 which carries the same identifier. This is a very widely used system today. Users order off the shelf, get uniquely coded products and use them for storage, and are absolutely done with the problem and risk associated to ID duplication. We also offer a service of customization, which also sometimes users take advantage of, but I think could suggest that mostly the unique off-the-shelf production of tubes is what is asked for. Exemplified here in these little pictures is data metrics codes, which often, or in every way actually, are on the bottom of the tubes. And then we see linear barcodes on the sides of the tubes exemplified here. You should know, and this is becoming also a little bit a topic on the next slides, that these, since they are on the bottom, and if you show an entire array of tubes, you can see them all at once. You can see 96 codes at once. They are often used for reg scanning, for scanning a whole bulk of tubes at once, and then you know of a batch of 96 tubes, what were the identifiers, where were they in the reg, what was their position, and you can very quickly process these tubes in your workflow. But at other stages in the workflow, it may be a requirement to identify individual tubes, and for this purpose also data metrics codes can be used, but often also linear barcodes, which are on the side of the tubes. Here, a bit more in detail, these features are shown. A smaller tube, a small volume biobanking tube with its two-dimensional code on the bottom, and here a 2 milliliter larger tube with its linear barcode and human readable feature. Also, the small one has a human readable feature here. And the point I want to make with this slide is that you find identical functions on regs, because you also want to trace your samples on the reg level. This gives you redundancy and also different options in automation, and you want both. Linear barcodes are on the side of the regs. They are in a standard position so that the automates can handle them, and they have a human readable feature underneath. Same also for larger regs. And also we have 2D codes on the reg bottom, which allow for the bulk scanning, and not only of the tubes, but the reg also in addition in the same run. And of course, I guess it's not important to say that all the different features, the linear barcodes, the human readable functions, and data metrics always encode or show the same identifier. Yeah, whatever you would scan or read, it would be the same unique ID. So yeah, again, just briefly, always think about what you need. Is single scanning what you need in your workflow, or at least at certain stages? Is bulk scanning, reg scanning what you need, or is human reading what you need? And then make a choice of Labware, which has the features this would require. Going back to the workflow, I want to emphasize one last time the concept of different reading functions which may play different roles in the workflow. Single scanning is very often applied and often during the entire workflow. And we see a lot of workflows today where reg scanning is also the case. But just to substantiate once more what I said before, human reading with the bare eye is sometimes getting of importance, and you should be prepared for this function. The example I usually make is, let's assume you have a very nice biobank, you have like a web page where one can access your data bank base, and there's very interesting samples, and there's a researcher working on a certain topic requesting 3 of these samples somewhere in the world. They get them shipped and they get paperwork with it, but these people usually don't have the single tube or reg scanners or whatever to really cross-check if really the right samples with the right paperwork were shipped, but they have their eyes, which they can always use. So the human readable function may at least become important at this very end of the workflow. I want to finish with these tracing and tracking features by coming up with 4 or 5 best practices in this context. And I would strongly suggest that machine readable codes, barcodes, data metrics codes should be used. Codes should be an integral part of the tube or plate. They should not be applied by adhesive labels. Adhesive labels at a first glance are not a bad solution, but it's really after years of use in the field, really problems have been reported. They are just not as durable if it comes to a long period of time or very low temperatures. Think about mechanical, chemical and temperature resistance, which you require, and then make a good choice. Lasered barcodes on tubes are often the best choice here. Code quality should be high, and this is simple because these codes have intrinsic error correction functions. One of them is illustrated down here. It's the Reed-Solomon error correction and data metrics codes. And what has been done here is a barcode, a data metrics code, has been replicated, has been printed 5 times, and then with a very harsh, super hot needle been destroyed on purpose. And what you see, it tolerates quite some damage. But this tolerance, this error correction, you want to save for the unlikely event of a sample getting damaged during its lifetime, but not you want to compromise data metrics quality from the beginning on. Yeah, that's the point. So we heard a little bit about automation, and you have even seen these little videos where full automation concerning the capping and capping process was shown. I just want to round up this picture a little bit with this slide. This is pretty much to sharpen the awareness. Of course, for many users, this is maybe already a common knowledge and common understanding. But it's worth to reflect on the fact that a lot of efforts is done today, and a lot of focus is put on the duration of processing of a sample in your workflow. So basically from the step of collecting a sample until the step of making sure the sample is safe, which often means it's frozen and it's stored in the cold. We know today that sometimes this can take really long because you get samples from the outside and there's shipment involved and all these kinds of things. But the more centralized everything and the more you can get control on the things, the more there's the suggestion that the gold standard is actually 30 minutes from drawing a sample until freezing a sample down. And this is a very, very short period of time. But again, it's the gold standard to best preserve biomarkers. And if this is the gold standard, and even if you centralize, you really have to think twice what all you can do, and then automation comes into the play, of course. And it also has to do with throughput of samples. The idea is that already decades ago in the high throughput screening field and world, standards have been developed, and one of them is the SLAS ANSI standard, former SBS standard, plate footprint standard. All our microtiter plates follow this standard, and also these 96-way rakes and also 48 format rakes follow these standards. And if you decide to use these standard rakes, you are definitely prepared for a whole range of automated processes you can carry out because, and this is of course not a secret, the automation partners have the equipment for these steps. And it's also worth to think twice if you maybe want to store in such standard regs, even if you don't automate today, because your project may increase, your biobank and store may get larger, you may get higher throughput of samples. And then you are prepared for the future. You have your standard Labware material, and you can use it today for manual handling or semi-automated, and you are future-proof. To change a Labware in the future and then have still your old stock, which you don't know how to deal with it, is always causing a little bit of trouble. So if automation is maybe at least a concept you want to use in the future, think about using such standardized reg formats already today. I just listed on the right-hand side what all is possible. We have seen the tube picking and capping, decapping, pipetting, liquid handling is possible, aliquotation, but whole derivatization like RNA, DNA extraction is possible. Even PBMC isolation is possible. And also in this store, it doesn't end. In the store, exemplified here, a Hamilton Bio storage system, these large freezers, so to say, there is still automation going on at the inside. inside of the devices. Samples are retrieved, racks are semi-full, half empty, and then you can do the condensation. All this can be done automatically. It's really fantastic what is possible today, but it all goes back to automation-friendly Labware formats. And with this, I want to enter the last stage of last phase of my presentation, which is now dealing a little bit with quality certificates and contaminants and also leachables. I want to start with the biological contaminants. And I want to, let's say, educate or give some insight here a little bit what actually manufacturers do to get control over these items, or over these biological contaminants. It all starts with hygiene concepts in production, clean room production, workers wearing lab coats and gloves and protect their mouth and their hair and all these kinds of things. So this is what comes first. The idea is never to produce something that may be a little bit contaminated and clean it afterwards. The idea is avoid contaminants, and this is what is state of the art and good practice in manufacturing today. And this of course only goes together if then also a monitoring step is carried out with a certain frequency, several times per year, where you really say, okay, I did my prevention of contaminants and everything, I monitored my clean room and everything, but I also need to show that all this of course is effective. Yeah, these two things in combination, one after the other, allow for a manufacturer to produce tubes that can be claimed free of endotoxins, free of DNA, and so on. Greiner Bio1 calls these tubes non-sterile because sterile is a term that requires something different, which we will see in a minute sometimes, but also you find these tubes sold under the term production sterile. But real sterile tubes, and this is norms which tell us when can we use this term, require something different. And this is destroying contaminants which still may be there. So you try to avoid them, but you assume still there might be something there. Now I destroy it on purpose. And this is usually done by using irradiation. It can be gamma irradiation, it can be E-beam or X-ray. For our production, we use E-beam, which is a very gentle method, because it doesn't do much harm to polymers. And then there's also a security level provided, and everything ends finally with a certificate and a validated process, because there's an ISO norm and you validate your process against this ISO norm. So if you buy non-sterile or production sterile tubes, they are usually certified, and there's a reason for it, free of DNA, RNAs, DNAs, and detectable endotoxins, and some other things sometimes as well. But this is basically the standard assortment of claims. And then if you order a product which is sold as really sterile, then you get in addition the sterility claim, and you get a sterility assurance level. You find two different on the market, 10 to the -3, 10 to the -6. 10 to the -6 is the gold standard for medical products, and we also apply it for our biobanking and cryotubes. This was biological contaminants, but there's also chemical contaminants which may be present in tubes, and you should at least be aware of the fact that there is something and something could go on. Polymers usually contain different chemical substances to really allow for their processing and in injection molding and production lines. There is, though, a huge variety of different substances you find or you do not find, and also the amount varies really extensively. Some of them are listed here. I don't want to really read through them, but mold release agent is one. We will see in a minute what this is. UV stabilizers, you can imagine, and so on. So these substances are sometimes present in the polymer. But it does absolutely not mean that all this stuff is extracted and really to be found in your stored sample. This is absolutely not the case. And the whole science around this discriminates between leachables and extractables. And what we talk about is leachables, because leachables is everything that could get out or gets out under regular use conditions. So the entire leachable topic, when we now talk about it for a few minutes, needs to be now considered in the context of I'm storing samples at room temperature or preferably in the cold. I'm not boiling a sample, I'm not doing whatever, I'm storing samples and I preserve them. And the idea is that extraction is performed under regular use conditions with solvents, and then GCMS or HPLC is used to check if there are certain peaks in the extract containing a leachable versus the control. And if this is the case, then you can of course start to check what is it, can this harm my process? I provide this insight, but on the other hand side, I have to say at the same time that today we have no overarching guidelines or standards for leachable assessment, in particular not for biobanking. Even in medical devices, the whole science and understanding is not really very advanced, but we also see that there's a lot of discussion and the world is changing. I would not be surprised if soon we would have better regulation which allow and standards which allow for a good understanding what is in these products and what could leach out. But even if I say this, I want to emphasize that today there are some guidelines and standards out there which can be applied, which sometimes are applied, and an end user can ask for them and can see what suppliers respond. Some of them are more common and more state-of-the-art, like heavy metal-free statements, phthalate-free statements, TSE, BSE-free statements. But if it comes to mold release agents and then USP Class 6 medical grade, this is definitely not a standard today. We suggest that it could though be applied because it's a very good thing that exists, and there are these guidelines. Why not to use them? I will not have the time to really go absolutely into detail here, but I want to very quickly talk about 3 of these items which we as Greiner 1 believe they are very meaningful and could and should be applied. One is mold release agents. These are agents that can be used to release the injection molded product, storage tube or plate from the injection mold. Sometimes this is a tricky process. Mold release agents can absolutely facilitate and help here, but they can be avoided. The whole problem can solely be solved technically, and we believe this should be the case for storage vessels because these substances can be avoided and you shouldn't find them in your sample at the end. Then there's medical grade polymers. Here the idea is also certain cleanliness. It's especially about batch to batch variation. You don't want contamination with other raw material types. If you ask for polypropylene, you don't want this to be mixed with the polystyrene. But what is also behind is that you actually get a better contract with your material supplier. Material suppliers cannot discontinue material with very short notice. They have to give the supplier time to find an alternative. And if you want to find a high quality polymer, this can take some time. There's a USP Class 6 statement, which is actually anchored in the US Pharmacopoeia. It is regarded the highest class of certifications you can find today for materials for medical applications. We do not necessarily have a medical application here, but what is good in medical applications, implantation and whatever, can definitely not harm the storage and biobanking world. Tests comprise several levels of toxicity. Some of the tests are done in vivo. And this, if a material is certified USP Class 6 and frequently tested, this is a very, very good statement related to biocompatibility and also cleanliness of the material. And last but not least, there is a similar statement which is more related on in vitro tests. It's the ISO 10993. It's section 5. This is also a standard which can be applied today, and what you usually find on the certificate is our product is cytotoxicity or is non-cytotoxic. This is how I have to put it. At Greiner Bio 1 today we apply all these 4 standards and rules for our products. I want to end with yet a different quality mark. You can find sometimes out there. Some products are certified. Again, I'm proud to suggest and that I can tell you that our products fulfill this criterion. And this is the CE IVD mark. Our storage tubes, if it's the larger or smaller ones, they carry the CE IVD mark, and Actually, they are also FDA listed as a medical product. If you do it in one country, you usually also do it in other markets. And this qualifies our storage tubes actually for use for storage of samples, which later on are meant to be used in diagnostics. And there's also a lot of discussion going on, how will the regulation around biobanking look in the future? Because what often you actually do or base on your samples is pretty much a diagnosis, and in the future it may definitely be, or you develop a diagnostic tool. And in the future we believe also these processes will fall into this context. So we suggest the ISO 13485, which is the norm behind it, which is applied, guarantees for two things. You are safe for the future if you one day go into more diagnostic applications, if it's really diagnosis or development of diagnostic tools, and it's just a very good quality seal you can apply today. I want to emphasize 2-3 things what are happening if a product is developed and marketed which has the CE IVD mark. You first of all have to design the product according to the rules which also apply for medical products, and you have to do a risk assessment which you would also need to do for medical products. And then post-market, if products are developed and sold and launched into the market, then you have to do post-market surveillance. That means we as a Greiner Bio1, for instance, we have to really every year check all the public databases. We have to go to the conferences, and we have to consider every knowledge that we can get to check, is our product still safe or not? And if there's any finding somewhere with cryogenic tubes, we have to assess if this applies to us, how the risk is for our product associated to it, and then act accordingly and come up with measures to correct for it. I want to finish with the question, how can you actually see if a product is CE IVD marked? You can see this on many documents and also the web page and everything. But the best thing is the literature of the manufacturer, its intended use statement, and last not least, the CE symbol that you have that you find on the label or even on the product. And it's also a must. If a product is a CE IVD mark product, it has to be endowed with this symbol, as it's shown here on this exemplified label. So I want to conclude with the open question: what makes a good storage container? Remembering everything I tried to emphasize in my presentation today, I suggest, it's not a very definite and clear answer. It must be just fit for purpose, for intended use, and for workflow. But very often this means that an appropriate and often wide temperature range shall be covered, and products must be suitable. Also, tracking features must be suitable and must exist. There must be automation compatible, which often is important. There should be suitable closure systems. Products must be proven free of biological and contaminants, chemical contaminants, at least concerning the state of the art. And often, and I didn't have the time to talk about it today, it is good that a storage container is also compact and space optimized, because what you don't want to do is to waste storage space because you have big plastic containers with little lumen and volume at the inside. What you want to do is store a sample, and the container is just a thing you need and can't avoid, but this shouldn't take up all your volume. With this, I want to finish, and I hope that we got some questions, and I would hand it over back to James for some questions.
Thank you, Sven. That was very interesting. And yes, we do have a few questions. I think we can probably look at 3 or so, depending on how long we've got. The first question is, I currently flash freeze my tubes in liquid nitrogen as part of my current workflow. Can I do this with coated tubes?
Yes and no. The thing is, flash freezing tubes is usually against the intended use of tubes, because there is the risk of liquid nitrogen entering the tubes And if you would take them out later on, they could really explode and break and even be harmful to people. So the flash freezing is always a risk, which is, I'm sorry to say this, but it's really the common practice, which is risk. The risk is on the user's side. I understand that sometimes it cannot be avoided, but this is simply a thing one needs to know. Be super, super careful, wear goggles and everything in case something happens. If it comes to the just product features like the lasered barcodes and all this kind of stuff, they will be stable, they will not fall off. This would not be the problem.
Okay, I think that's an excellent answer, Sven. Let's have a look at another one. We are currently looking at whole process system tracking. Can storage plates be barcoded in a similar manner to the vials?
So in storage plates, it also applies for our high throughput screening plates. We have options for linear barcodes on the side. We would probably need to go in detail to understand which plate exactly, which barcode format, and so on. So we could consider it, absolutely. We can do it. We can, though, not endow these plates with any data matrix codes or lasered codes today.
Okay, that's good. Moving on. We have another one here. I'm very interested in the small barcoded tubes, but at the moment, part of our process is based on coloured caps using 1 and 2 mil tubes, based upon groups within our floor. Can you have coloured caps for the 96 position small tubes, or is it inserts?
So here I can proudly announce that the color variety of our small volume tubes is the largest we provide in our assortment. Yes, we have them. We have, I think it's 10 different colors or something in that range. We have a lot, and they can absolutely be used for this purpose. Yep.
Okay. And we have one here which may or may not be able to answer it. It's a later one. Here we go. Will the barcodes be affected if they come into contact with harsh chemicals?
So we provide a statement. We have tested all kinds of different chemicals, like the most common ones, starting with ethanol, but then of course also DMSO, which is used in compound storage or cell storage. So we have these data available. I have to say, and that's maybe not a surprise, the barcodes are not resistant to everything like forever exposure and whatsoever, but they are resistant to the most commonly used chemicals, which is ethanol, isopropanol, DMSO for sure. And if one wants to go further in detail, we would provide our documentation and the test results we have, and then we can check which chemical is of interest and see how robust it is.
Okay, that's a good answer. That also answers a previous question about ethanol as well. And then I think if we go with one last one, when scanning the racks you mentioned, can you see where in the rack the tube is? I think that's referring really to rack scanning. Yeah,
today we even provide the service, everything that is pre-racked. So the tubes are in the racks. They are in an order. So you could even in theory say, I have rack number so and so. Which tube ID would I need to expect in the first well, like A1 or so, if I just get product from the supplier and haven't changed the order? So even this is given the case. And then if it comes back to can I link it? I mean, I hope I understand the question right, but what you would do is you scan your rack. You scan, for instance, 96 tubes, and as a 97th code, you scan your rack ID on the bottom, so you have 96 codes. And then in the software, of course, all the different tubes are linked to the rack code and also to their position where they were found. So absolutely, yes, this is the case.
Okay, that's wonderful. And then if we make this the last one, I would like to know if there's any difference between the external and internal screw caps on the larger cryovials.
On the larger cryovials.
Quite a broad question, this one.
Yeah, I mean, yeah, this is, at least for Greiner by one, it's a rather historically grown portfolio. We started many decades ago with one, and now we have both of them. And the pure reason is that people just prefer one or the other, and sometimes it's almost like personal preferences. If you look at it in a more rational way and say, okay, what if it comes to features and so on, they both are the same. We guarantee the IATA, the air freight statement. We guarantee tightness. We guarantee that they can be used in automated systems. So pretty much it's the same. We see that sometimes there was a bit a preference for the externally threaded tubes if they were more handled manually, because there's more of a gripping surface. And then the automation partners like the internally threaded tubes better because it's easier to cap and uncap them. So there's a slight little difference if it comes to automation and also if it comes to more manual handling. But function-wise, they are pretty much the same.
Okay, that's excellent. I think at this point we'll bring things to a close. Any questions that we have missed, and there is a few, we'll try and come back to those offline. And if anyone wishes to get in contact, follow the Greiner's normal page and also the contact details at the end of Sven's presentation. Well, thank you very much, everyone, for joining us, and have a good remaining of the day. Thank you.
Thank you, James. Bye-bye.